Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more of the authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is more of an open format discussion of additional articles, conferences, or major events in the field that we'd like to highlight. My name is Darleen Sandoval, and I am hosted by my podcast co-hosts, Dr. Kirk Habiger and Dr. Kevin Williams. Hey guys, how are you doing today?
Hey, Dar. How's it going, Kevin?
Doing good.
So we're in the full swing of fall. So I'm gonna put you guys on the spot, and I'll go first so you guys can think about your answer. But what is the seasonal event that makes you think, oh, it's fall's coming, fall's here? So for me, I'm from New Mexico, and so it's definitely the smell of roasting green chilies in the air. Kevin, how about you?
It's a good question. I'm going to move on to Kirk right now.
I've been in the South for over a decade now, and fall is much later and very short, but it's that weird day where you open the door and the humidity doesn't jump down your throat.
Nice.
Yeah, it's a wonderful feeling. So
Yeah. Do you get much color in Birmingham?
we do
Fall
get a
color?
little bit. It is
Yeah.
it's rather quick before it goes from color to on the
Yeah.
ground.
Yeah. What about you, Kevin? Did you come up with something?
I think I have to say football season.
Yeah,
That's when
a
I
good one.
know fall is coming. When
Yep.
you get out of preseason and you're getting into the regular season games. Being from Dallas, I know I probably get a lot of hatred for being a fan of Dallas Cowboys. I grew up in Dallas, so I have to be a fan of them. I wish they would be doing better, but yeah. So
All right, that's awesome. So I guess we should get to work. Kirk, why don't you tell us what we have in stock for our listeners today?
Yeah, so today we are joined by lead and co-corresponding author, Dr. Magnus Grøndahl, Faculty of Health and Medical Sciences, and Doctor of Medicine at the University of Copenhagen. He'll be talking to us about his paper, Glucagon Clearance is Decreased in Chronic Kidney Disease, but Preserved in Liver Cirrhosis. Then we get a chance to chat with Dr. Jessie Barra, lead author and postdoctoral fellow, and her mentor, Dr. Holger Russ, associate professor at the University of Florida, about their paper, Cryopreservation of Stem Cell Derived Beta-like Cells Enriches for Insulin-Producing Cells with Improved Function.
All right, awesome. And Kevin, what do we have in store for our Sweet Talk segment?
today we welcome Dr. Siddharth Sunilkumar and Dr. Michael Dennis into the Sweet Talk segment. They will be discussing research funded by the ADA Pathway to Stop Diabetes Initiator Award in 2014 that was awarded to Dr. Dennis. This work focuses on mechanisms behind hyperglycemia-induced changes in blood vessel growth of the retina, with the ultimate goal of developing new treatments for diabetic retinopathy. And we were discussing this earlier, and I believe this is the first time we are featuring a segment on the podcast on diabetic retinopathy, which is really such an important and relevant topic. And while we didn't plan this, I recently found out that October is the National Vision Awareness Month. So this topic is very timely and a reminder for everyone to take care of your vision. I'm sure our listeners will enjoy the discussion with Dr. Sunilkumar and Dr. Dennis later in the program, so stick around.
Kevin, you absolutely planned that. You should not, you should absolutely be taking credit for that. It was not by accident.
Yep.
All right, let's jump into it.
Now, I want to alert you to a special offer. The American Diabetes Association is extending a special offer for new and renewing members. When you sign up for a professional membership to the ADA, use the code FALL24, that's FALL24, during checkout. And this will get you 15% off the yearly membership price. And if you sign up for a 2-year membership, you'll receive a 15% discount and a free It Takes All of Us t-shirt from the ADA. In addition, you will get exclusive discounts on ADA's scientific sessions and other meetings, a subscription to one of ADA's journals, a copy of ADA's Standard of Care in Diabetes, and other membership exclusive discounts. Again, that's 15% off a 1 or 2-year ADA membership using code FALL24 in all caps, that's FALL24 at checkout. and the offer expires November 30th, 2024. Don't miss out on this fantastic opportunity to expand your commitment in the fight to end diabetes by joining the American Diabetes Association.
For our first paper of this month's podcast, we're going to discuss a feature article titled Glucagon Clearance is Decreased in Chronic Kidney Disease, but Preserved in Liver Cirrhosis. We are joined by the lead author, Magnus Grøndahl, who is a PhD student at the University of Copenhagen. Welcome, Magnus. Congratulations on your paper, and thanks so much for being a part of this month's podcast.
Thank you. Thank you for having me.
Magnus, it's great to have you here. And we always like to start with an icebreaker question, right? So we didn't provide you any of these, and it has nothing to do with your paper, but
Right.
maybe you can give us a fun fact about yourself that has nothing to do with science, or just gives us a little insight.
Oh, nothing to do with science. Well, I think a lot of my time these days goes with science, but I like playing basketball, and I think that's probably the closest connection I get to go to with you guys in the States, that I like watching American basketball. So yeah, that's probably a fun fact about me.
Yeah, that's a good one.
Do you have a favorite team?
Celtics. So I'm happy these days.
All right. Well, first, we like to give our authors the opportunity to highlight any other contributing authors or collaborators to this work. Do you have anybody you'd like to acknowledge?
Yeah, I mean, this work wouldn't have been possible without all the recruiting clinicians. So I do my research at an endocrinology research department, but this piece was mainly done on kidney patients and patients with liver cirrhosis. So all the recruiting clinicians who actually saw the patients, saw that they were eligible for this study, they deserve huge credit, because otherwise we couldn't have recruited all these patients.
Awesome. And then you are currently, at least for a couple more weeks, correct, in the laboratory of Dr. Philippe Knopp.
Yes.
Can you tell us a little bit about his body of work, just sort of introduce the listeners to what he does and maybe a little bit about what led to this project?
Yes, so I've done my PhD at Center for Clinical Metabolic Research, and the name kind of gives it away that we do clinical research on both people with various metabolic diseases, such as type 2 diabetes, type 1 diabetes, obesity, and also healthy control individuals, both to gain more insight into the disease, the pathophysiology, but also just the basic physiology of humans. So we do infusion studies with all the different gastrointestinal hormones that you can think about. Of course, glucagon like this one, and insulin and GLP-1, but also the more rare like GLP-2 or LEAP-2. And we also do intervention studies, meal tests, and obtain all kinds of biopsy materials from people undergoing different kinds of metabolic surgeries. So we're really passionate about all the endocrinologies, the metabolic pathways, and different kind of endocrine diseases.
Awesome.
I want to jump into the article a little bit here.
Yeah.
And I want to start with the experimental groups that you looked at, right? You chose groups of individuals with Child-Pugh cirrhosis, right?
Yes.
Versus those with chronic kidney disease. Maybe start out with explain a little bit what Child-Pugh cirrhosis, what the phenotype of those individuals is, right? This is a specific liver disease, right? And then why did you choose to compare those two groups as compared to the control group then?
So we wanted to have some models where there were singular organ impairment, either liver impairment or kidney impairment. And we wanted to be chronic and not acute, so we could study a more stable phase. And we came across cirrhosis, I think because it was this stable model of chronically decreased liver function. Ultimately, we wanted the patients with most severely affected livers, but the Child-Pugh C was probably too affected to participate in this kind of study. So we ended up with the A and B. And that phenotype is, well, at first we didn't thought that it would be that comparable to the chronic kidney disease patients, but as it turns out, they were actually quite comparable in terms of age and demographics and stuff. So these patients were mainly diseased in their liver due to alcohol intake. So it was like alcohol-induced liver cirrhosis. So we wanted to see, we could fairly see in there when we analyzed their liver that they were quite liver affected or had a clinical impairment in the liver function. So we thought that would be a nice model for this setup.
Right. Thanks. And then investing glucagon in the context of chronic kidney diseases, right? I
Mhm.
think that we're all gaining an emerging appreciation for its role there, but maybe you could give us a little more insight into why you chose that group as well.
Yeah, we did so because we and others in the literature think that the kidneys are the primary organ for glucagon clearance, and that glucagon is playing a role in the pathogenesis of type 2 diabetes and diabetic nephropathy. So we wanted to have a again stable cohort of patients with markedly decreased kidney disease. We read some articles showing a nice inverse relationship between kidney function and fasting glucagon levels, published a couple of years back. So we really wanted to try to dig deeper into that. And there were some studies done in the 70s on patients with chronic kidney disease as well, but with analytical techniques. So we wanted to try to reproduce those findings from those older studies with more modern laboratory techniques.
Thanks.
So with that, let's talk about what you actually did. So you measured basically your primary endpoint is the metabolic clearance rate of glucagon. So can you tell us a little bit about how you did that and what you think that means in terms of the biology or maybe even the pathobiology?
Yeah, so you can describe the elimination kinetics of hormones of glucagon of any kind of substance using different kinds of parameters. And we chose the metabolic clearance rate to be our primary endpoint, as it gives an overview of how much glucagon is cleared per minute from the body, and then standardized to body weight or surface area, in this case body weight. So it's in the same area as half-life, and we also described half-life. So it gives some measure of how well your body eliminates glucagon per minute time. And we evaluated it by infusing glucagon intravenously into these three groups: patients with chronic kidney disease, patients with liver disease, and healthy match controls, and then measuring their glucagon levels before, during, and after the infusion, meticulously every 2 to 5 minutes, to see the rise and the stable concentrations and then the decay of glucagon again, in order to calculate these kinetic parameters.
And it seems like so you also paired this with the glucose tracer. So tell us a little bit about that. And then I guess maybe as a tag on to the metabolic clearance rate, you also could have labeled the glucagon, correct? Is there a reason why you chose metabolic clearance rate rather than labeling the glucagon?
I think it was a technical aspect that
Yeah.
we know how to do this. We've done it before. We did this in patients with type 2 diabetes before this one. That was the first study of my PhD. So it was easy. We knew this setup. We don't have a lot of experience with the glucagon tracer, although that will be very interesting to try to apply in these settings. And the glucose tracer was actually also a realization that we did in the first study with the patients with type 2 diabetes, that we really wanted to have a clearer insight into their glucose homeostasis. So we added the glucose tracer to maybe see is there a difference in the rate of appearance and the endogenous glucose production that we cannot see just by measuring the plasma glucose alone.
Did you think that there might be some sort of glucagon resistance, if you will, in these
Yeah,
patients?
perhaps.
Yeah.
We talk a lot about glucagon resistance on the amino acid level in our group and in our research. Steatosis-induced glucagon resistance, and in this case, I suspect that the patients with liver disease would demonstrate a decreased glucagon response, at least on the glucose production. That was my hypothesis to begin with. So that's why we did it, because they were also characterized by the increased insulin resistance, and that might have balanced out that weaker hepatic glucagon response. That's why we did the glucose tracer.
Yeah, so that's a great introduction to my next question, right? Which is, tell us a little bit about your results, right? Highlight the differences between the chronic kidney and the chronic liver disease. And were you surprised by any of these results, right?
Yeah. So one of the reasons we did this study was that the literature before our study was kind of in conflict with itself, whether or not the kidneys or the liver were too much, were the primary organs of glucagon clearance. So we tended to agree with those saying that it's the kidneys. And in our study, our main results is that, as the title gives away, that the glucagon clearance is decreased in kidney disease, but not in the chronic liver disease. Again, highlighting the kidneys as the main contributor, or one of the main contributors, at least more than the liver in glucagon clearance. So that's our main takeaway from this study. And that, of course, the negative result in itself that patients with liver cirrhosis have a preserved glucagon clearance. That's the main. And then we also measured different kinds of effects of glucagon, which were fairly similar between the three groups, which was, I think, surprising. Again, coming back to the part about glucagon resistance and the liver cirrhosis and the impaired liver function there. So I think that's the main takeaways from this.
One of the things that struck me as I was reading your paper was that if there's normal clearance in the cirrhosis group, right, but elevated glucagon, right, the kidneys are exposed to more glucagon, right? Did you see any evidence, or is the pathology of these diseases so profound that you can't really see excess glucagon action in the kidneys of the cirrhosis group?
No, we couldn't. We couldn't.
Okay.
I mean, and we didn't really have the tools, I think, to measure that other than urine output, which we measured, which was similar across the three groups. And there were perhaps some effects on the cardiovascular system on the liver cirrhosis group that wasn't present in the other two groups. But other than that, they were actually quite similar, surprisingly similar, at least for my part. Yeah.
Thanks.
Awesome. You have a nice part in your discussion where you talk about some of the weaknesses of the paper and, potential problems interpreting those results. And one of the interesting things that you noted was the potential confound of the insulin resistance in the cirrhosis papers. Can you talk a little bit more about that?
Yeah, so that's one of the main limitations, I think. We didn't clamp insulin in this study, and we wanted to interfere as little as possible with all the other metabolic pathways, which is why we didn't apply a pancreatic clamp with somatostatin. But that, of course, leaves us with the problem that they had differing insulin responses, and the liver cirrhosis group secreted more insulin, or at least there were more insulin in their bloodstream, and we calculated the insulin clearance, which is mostly hepatic, and that was decreased. And now we're talking insulin clearance instead of glucagon clearance, but that increased insulin could play a role, at least in the effects of the glucagon infusion, as that would counteract the glucagon infusion.
That's a great observation. So I guess at this point, are there any other key thoughts that you'd like to leave us with, or maybe give us a hint about where you hope this research will go in the future?
One of my main takeaways from this study as well is that it matters so much what kind of experimental setups and analytical methods that you use, because the discrepancy between analyzing glucagon with a radioimmunoassay and analyzing it with a sandwich ELISA, it was large in this study, and could also explain some of the discrepancies in previous studies and older studies. So I think that's a good takeaway from me, at least, that we also need to talk about the methods and not only the results.
So, Magnus, before we close, I want to give you the opportunity to talk about the fact that you're going to defend in a couple of weeks your thesis. Do you have a postdoc lined up? What's next for you?
I haven't a clear path to a postdoc right now, or that is maybe I do, but right now I'm transitioning back into the clinical work at the hospital.
Okay.
And so I've recently just begun at a pediatric department here in the greater Copenhagen. And I hope to combine the two, both the pediatrics and the endocrinology research in the future. Yeah.
Oh, awesome. We
Yeah.
definitely need more clinician scientists, so I'm excited to hear that.
Yeah.
Well, thank you, Magnus, so much for joining us today and sharing your expertise, and for discussing your recent work on glucagon clearance rates in cirrhosis and chronic kidney disease. I encourage the listeners to read the full article in the October issue of Diabetes.
Thank you.
Up next, we'll be discussing a featured article from the October 2024 issue of Diabetes titled Cryopreservation of Stem Cell Derived Beta-like Cells Enriches for Insulin-Producing Cells with Improved Function. Today, we are joined by the lead author of the study, Dr. Jessie Barra, and the corresponding author, Dr. Holger Russ. Dr. Barra is a postdoctoral fellow in Dr. Russ's lab, and Dr. Russ is an associate professor at the University of Florida in the Department of Pharmacology and Therapeutics. Welcome, Dr. Barra and Dr. Russ, and congratulations on your paper.
Thank you.
Thank you very much. Glad to be here.
So when we start these podcasts, we usually start with an icebreaker question that we didn't provide ahead of time. And so I should note that Jessie, Kirk is very sad to not be here. He had a scheduling conflict. But usually we give you guys the same question, but because of the insider information with Kirk, you get a separate question. So Kirk wants to know, he says, you always drank coffee with your graduate student friend in the corner of the hallway. And he wants to know, where do you drink your coffee now? And have you found a new caffeine enabler?
Oh boy. Usually, sadly, I don't have a fun answer for this. I drink my coffee at my desk now.
Ah.
But no, the caffeine intake is extremely important still.
Yes.
There's a lot hidden in that question.
Yes.
Yeah, we used to get that we would waste a bunch of time, but we really weren't. We were actually discussing a collaborative project that ended up being a paper. So it was productive time.
Oh,
Good,
awesome.
good. All right, that's awesome. All right, and Holger, we'd like to know from you a fun fact about yourself that has nothing to do with science.
Fun fact, in the few times I take off, I like to go chase waves. I like surfing a
Oh.
lot, and
Nice.
Wow.
I don't get to do it as much as I should, but when I do, I enjoy tremendously.
Oh, that's awesome. All right, let's get started.
Dr. Russ, first, we like to give our authors the opportunity to highlight any other collaborators or authors that may have contributed to this work.
Yeah, of course. Want to thank the whole team. My lab basically had University of Florida, but also my postdoc and former grad student, Roberto Castro-Guedes, who is now postdoc at UCSF in Tongue Lab. He was essential in the paper, as well as one of my grad students here, Austin Gratz, at UF. And then also want to highlight my longstanding collaboration with Dr. Ed Phelps, who is freshly minted associate professor in the Department of Bioengineering here in UF. I also would like to thank the funding agencies involved in basically helping us and allowing us to conduct the work and still doing this. And then we also would like to very much thank the organ donors and their families for allowing us to use the tissues for this important work that will ultimately help people in need, right, affected by the disease. So thank you very much for allowing us to work with these precious tissues.
Awesome. That's great. And wondering if you can give us or start us out with an overview of your research program and how this specific project fits into your research program.
Yeah, sure, gladly. So over the many years I'm working on the pancreas, right, I have been more and more interested in really interrogating the immune beta cell interface and doing this in a human context, right? And why this is so important when you think of it, many smart, hardworking people have researched autoimmune diabetes for decades, and we still don't know how and why the disease manifests, right? And basically, I have made it, if you want to put it like this, the goal of my career to enable us to get at this question in a human context and using and relying heavily on stem cell technology in combination with genome engineering. So can we make the disease-relevant tissues in a dish? Can we manipulate them? And by doing so, understand what actually triggers the disease? Because once you understand the problem, you actually can intelligently try to fix it. So this is also how this paper, in a sense, fits into the overall theme of the lab that we're doing.
Yeah, so tell us a little bit about that. You talk about in your introduction the challenges to the more conventional approaches of generating stem cell derived beta-like cells. So can you tell us about some of those challenges?
Yeah, I can speak to this. So I think Holger would entirely agree that making these cells is extremely expensive and very time consuming. And while the paper that we're discussing right now didn't necessarily make advances in the differentiation protocol to make these cells, I think the additional challenge that this paper does try to address is once these cells are made in the dish, it's very difficult to maintain them in culture for long periods of time. And so we wanted to develop a method to address that particular challenge. And that's what this particular manuscript tried to do.
I'm curious, maybe you can elaborate on what the specific methodology is in this study. It hasn't been described quite yet for our listeners. In addition to that, how did you arrive at that specific strategy for these cells? And were there any approaches that didn't work out that maybe weren't discussed in the paper that you might want to elaborate on?
Yeah, absolutely. So our group has published on this protocol a couple of times, but as a broad overview, we start with human pluripotent stem cells, either from patients or from embryonic sources. And we utilize a three-dimensional bioreactor system to keep these cells in suspension. And then over the course of 23 days, we take these three-dimensional pluripotent stem cell clusters, so to speak, and we force them down a developmental pathway that tries to mimic in a dish the developmental cues that these cells would receive in a developing embryo to generate these insulin-producing beta cells. And that whole process takes 23 days.
So the 23 days is just the developmental time frame for it to get to that certain stage? What's special about 23 days?
Yeah, so this is kind of standing on the shoulders of giants, so to speak, of the field, having kind of worked out which signaling pathways in the dish allow us to push these cells down developmental trajectories. And so our lab, with Holger's background expertise stemming from his postdoc and just continuing into his independent career, we figured out that that's the time frame required to make these stepwise developmental stages. And that's just how many days it takes us in a dish to replicate this embryonic development.
Yeah, given we would like it to be faster, but we cannot accelerate development to a point which we like. So we have to live with the 23 days. To put this also in context, this is a very elaborate and work-intense process that requires attention, like feeding the cells every day. So it's kind of elaborate. And then once the cells are generated, right, in a timed manner, now when you want to do co-cultures, there's really this real life challenge of, do we have access to other immune cells? Do we need to generate them? Which really increase the complexity of the experimental model system considerably. So the motivation was like, can we introduce a stopping point that we can align our more complex experiments in a manner, right? And then, of course, we're like, okay, if we could freeze them away and pull them out and plug and play different models together, that would be tremendous for us, but also for the field, right? Because we can then also ship them. It will ease collaboration and basically just accelerate the discovery process in our hands. And that's kind of like a theme, right? When you think of how do we have acceleration in discovery, it's very often driven by our technical abilities. With the discovery of the microscope, right, we could look at smaller structures. So for us, this really has been a game changer to be able to cryopreserve our cells. Now, the cherry on top was that we found ways to, when we thought them out, that they even enrich for the cell type we are interested in, which also results in an improved function, right? Maybe it was lucky, but then sometimes the hardworking man or woman gets lucky. To put it this way, now speaking to where there are challenges on the way, as you know, a paper is always the polished product. It's a long road to get there. So of course there were failures, right? In the beginning, we need to find out how do we dissociate the cells? How do we preserve them? We started off with, okay, we wanted to freeze down whole intact clusters, but with the equipment available to us, we weren't able, basically, when we would thaw them out, most of them would be dead and not workable. So perhaps essential, while very technical, what was critical is to gently single cell the differentiated SBCs at day 23, and then cryopreserve them in a high density with low volume of cryopreservation.
Nice.
Nice. And what were some key findings that convinced you that this method was viable and useful tool as an alternative from the traditional strategies for generating the cells, preserving the cells? What were some of those key findings?
Yeah, absolutely. So as Holger mentioned, it was just kind of fortuitous that when we were looking to analyze and compare the cells before cryopreservation and after cryopreservation, we found that the fraction of the cell population that we were working with after cryopreservation was actually enriched for our beta cell population, those insulin-producing cells. And what we found was absent after cryopreservation was actually the small contaminating progenitor population. That's really commonly seen in this differentiation process. But we found that the cryopreservation and thawing methodology actually removed this population, which is fantastic, not just for enriching for your cell population of interest, but also if we think further down the line, right, if we could utilize this in a clinical approach, you also don't want those cells present in a cell population you're putting into people, right? So it was a really nice, again, fortuitous side effect of this cryopreservation method. And in addition to that, also importantly for clinical therapeutics, as well as our own basic science investigations, we found that the post-cryopreservation population was more functional, meaning they responded better to high glucose concentrations than the clusters before cryopreservation. So I would say those are the two key essential findings from the paper.
I'm sure that it wasn't luck, it was all skill.
I would like to add to this, to add to the complexity. So what was kind of intriguing in the study, as Jessie pointed out, that we see greater insulin secretion in response to high glucose, which indicates that probably the maturation of this cell population is increased. However, what is also a kind of a side takeaway from the story is that when we look at classical markers for immature or mature beta cells, we actually find an increase in the immature markers, which contradicts the functional data that we obtained, right? Now, what that reminds me of, and maybe the field, is that a marker is only as good as it gets, right? And it doesn't indicate the function. It's a correlative marker. And so it kind of questions if the marker sets employed commonly as of now in the field of stem cell-derived beta cells actually necessarily indicative of the functional state of the cells we generate. Also, to be frank here, there's probably more work that needs to be done to really tease out the intrinsicies of this process and the cell types we are generating and comparing it to primary human beta cells from cadaveric organ donors, right?
Yeah.
So lots more to do.
Yeah, definitely. I'm going to jump ahead a little bit. And you had talked about in the discussion that you felt that this strategy provides an opportunity for the wider diabetes field, and your hope is that it would accelerate research discoveries. So Holger, in your grand vision of this, what do you see some of the advances that it might be able to speed up?
I mean, the hope and the idea is that others can adapt this approach for their own research, which will result in them being able to cryopreserve the cells. And I will get to this, why this is important. And more importantly, upon thawing, enrich for the cell types we want to study and deplete proliferative progenitor cells that might mask or confound results being obtained. Now, from a practical point of view, what it does to us, and I hope also to other research groups, is that while we would plan out experiments with the differentiation period of almost being a month, right, out for weeks in advance, now we can basically focus initially our efforts in generating frozen cell banks from independent differentiation experiments. And then in theory, a researcher in my lab can pull out one tube one day after another and just repeat experiments, get like either, yes, we proven or we disproven our hypothesis that we tested, and it's a game changer for us. And therefore, we also felt it is important, and in a sense, diabetes, with highlighting the paper, verified that, important for the field to know and to learn about it. And then also for the people listening here, you can always send Jessie or me an email, and we will try our best to help, because the devil is always in the detail.
Definitely.
Dr. Holger and Dr. Barra, we want to thank you for joining us today to share your expertise and discussing your recent work on a new strategy to generate and preserve stem cell-derived beta-like cells. I encourage the listeners to read the full article in the October issue of Diabetes. Welcome to another edition of Sweet Talk. I'm Kevin Williams.
I'm Kirk Habegger.
And I'm Darleen Sandoval.
Today, we welcome Dr. Siddharth Sunilkumar and Michael Dennis. Dr. Sunilkumar is a postdoctoral scholar, and Dr. Dennis is an associate professor at Penn State College of Medicine. They have contributed a perspectives article to the October edition of Diabetes based on work that was funded by the ADA Pathway to Stop Diabetes Initiator Award given to Dr. Dennis in 2014. The focus of this work was to identify and understand mechanisms regulating hyperglycemia-induced gene expression that controls blood vessel growth in the retina, with the goal of developing new therapies for diabetic retinopathy. Thank you both for joining us today.
Thank you for having us.
Thank you for having us.
Before we get into the science, let's find out a little bit more about you. When you're not in the lab, what is a hobby or activity you enjoy that might surprise people? Let's start with Dr. Sunilkumar, and then we'll move on to Dr. Dennis.
So I think of my kitchen as my lab at home, and I love to cook.
Nice.
My weekends when I'm not in the lab generally spend baking, and I'm also a trained classical singer. So
Oh,
I
wow.
do my sessions with my teacher back in India when I get the time, which is not much these days, but yeah, I do try and keep in touch with that.
Do you have a favorite dish that you like to cook?
I've been experimenting my recipe or perfecting my recipe now on trying to bake macarons. So
Oh, nice.
I would say that's my goal for this year.
I'm sure the whole lab benefits.
Into the lab, yeah.
Sometimes I do bring some goods in. Most of my baking isn't professional yet, so I still critique myself a lot. But when they do turn out good, I do bring them in.
Yeah, after this, Michael might be asking you to bring in a lot more. Michael, what about you?
You know, I think, like Sid, my time outside of the lab has become very limited lately. But I'd say once upon a time, I was a runner. I used to run a lot. That becomes tricky with kids and lab and all the things I juggle nowadays. But
Absolutely.
certainly still enjoy getting out there. And central Pennsylvania is particularly nice, you know, especially this time of year when the weather's really cooling off, and you know we have some fall foliage starting to happen, and it's nice to get out there every once in a while still.
I'm guessing lots of rolling hills out there as well,
Yes,
right?
yes, exactly. Yeah.
Those are fun. All right. Oh, I guess I get to start with the first question, and we're going to jump right into the fun science. As Kevin mentioned, a major focus of your work is on REDD1 and its role, I would say, in the development of complications associated with diabetes. Is that correct? Yeah. So why don't we start with Michael, and why don't you explain to us exactly what REDD1 is and how it functions in the body, maybe in terms of normal physiology, and then maybe a touch on the pathophysiology.
Yeah, so REDD1 is a protein that's made in cells of our body when they experience stress. And this can be something simple like hypoxia or DNA damage or endoplasmic reticulum stress that the cell needs to address, it needs to adapt to, right? In the short term, the cell makes REDD1, and that's a good thing because it reduces energy expenditure and it allows the cell to kind of adapt to some of that stress that may have occurred. And this is by like shutting down pro-growth signaling. And what normally happens is after just a few minutes, that REDD1 gets degraded. So it has a very short half-life. It gets made, and then it only sticks around for a few minutes. But in the context of diabetes, kind of what we see is that in cells of the pancreas and retina and kidney and a number of other places, REDD1 gets made and it kind of hangs out. It gets oxidized and it doesn't get degraded properly. So that actually then leads to a number of problems and kind of a failed adaptive response to that stress. And it leads to kind of chronic inflammation and chronic oxidative stress that then causes those diabetic complications.
So I guess, building off of that, Siddharth, can you maybe tell us how REDD1 in diabetic complications like retinopathy and neuropathy might be targeted? in a way that we can help prevent or at least treat those complications?
That's a really good question. And so, like Dr. Dennis said, RED1 in diabetic complications, there's this continuous buildup of RED1, and there's a failure of degradation of that protein. In the last decade, there was a clinical trial that tried to target RED1 mRNA synthesis, and that was partially successful in mitigating vision dysfunction in diabetic patients with macular edema. So one of the ways to target it would be to suppress its synthesis itself by using siRNA therapeutics in the context of retinopathy. In the context of nephropathy, that has certain challenges in targeting specific cell types. And I think that's where the future direction of the research is in our lab as well.
Just out of curiosity, does it spill over into the systemic circulation, or is it really just kind of staying in the cell and causing problems?
So we haven't particularly looked at systemic circulation of RED1, and I think from my understanding, it's not a circulating protein.
Yeah, it's not secreted protein.
It's not a secreted protein. So I don't think it's a cytokine or a molecule that could potentially be used as a biomarker, if that's what you were getting at.
Yeah.
And building off that a little bit, Dr. Dennis, I was curious about those potential clinical strategies that have been tried or maybe that are in the future to be planned. The mention of the RNAi or the siRNA strategy and the knockdown of RED1. Is that the way the strategy should be for clinical development? Is there a strategy for the knockdown versus an inhibitor approach? Which one would be the most beneficial without causing potential side effects or harm? And this may be relevant because in your article, y'all do bring up the paradox of inhibiting RED1 can also potentially inhibit insulin signaling by upregulating mTORC targets. So is there a sweet spot for that inhibition? Essentially, how should these therapeutic strategies navigate the challenge?
This has been over a decade ago when we saw intravitreal administration of an siRNA targeting the RED1 mRNA that was given to patients with diabetic macular edema. And there was some modest improvement in best corrected visual acuity in those patients. So I would categorize those results as hopeful. But I think what was disappointing is that there was really no improvement in permeability. So the vessels in the retina still became very leaky. And ultimately, it was viewed as kind of being an inferior strategy to blocking signaling of this proangiogenic cytokine known as vascular endothelial growth factor. And over the last decade, we've seen that anti-VEGF therapies being administered intraocularly are kind of like the frontline treatment for not only patients with diabetic retinal disease, but for a number of other retinal diseases that are vascular in nature. What we know about siRNAs is that they cause the mRNA to get degraded. And what that does is it causes you to make less of a certain protein that is encoded by that mRNA, right? So that therapeutic approach to knocking down RED1 in the retina was trying to prevent how much RED1 was being made. But what we've recently learned is that the way diabetes works and the way diabetes causes there to be a problem with RED1 is that it prevents that normally rapid degradation of RED1 from occurring. So trying to reduce the amount of RED1 getting made is probably helpful in these patients, but it may not be an ideal therapeutic strategy for preventing the accumulation of RED1 that is occurring due to a block in its normally rapid degradation, right? So one of the approaches that we're pursuing now is to try to restore that normally rapid degradation. So to do this, we're trying to use small molecule inhibitors that would prevent the effect of diabetes on RED1 and allow it to still get degraded very rapidly. So that way, obviously that when there are stresses in our cells, we can still make RED1 acutely, and RED1 can have potentially those beneficial effects, but it doesn't become oxidized and stick around and potentially cause some of those chronic problems that we associate with diabetes. So we're hopeful that that's a better strategy for preventing the increase in RED1 in the context of diabetes.
So, Siddharth, to kind of follow up on that topic, it seems like a protein that it's not just, I mean, your focus is on the eye, but it sounds like it's a problem in other tissues too, including the islets, right? And so do these small molecules have a systemic effect, or are you still trying to target the eye in these strategies?
So I think that's something that we do address in this article as well, is that REDD1 is upregulated in multiple organs, but it also has a physiological function. So I think the potential challenge going forward would be targeting the expression and the abundance of this protein in specific cell types, thereby preventing the buildup of the protein in those cell types, and then looking to see if that's protective against the progression of the disease, and especially the progression of complications associated with diabetes.
Yeah.
So, for example, in the case of diabetic retinopathy, we have been able to show that RED1 buildup in a glial cell type within the retina is actually what leads to the progression of DR and the development of DR, at least in the preclinical models that we've used. So maybe targeting RED1 in specific cell types would be the way to go forward instead of systemically suppressing its expression, where in certain cells, it has a physiological function, which may be protective against diabetes as well.
I think you mentioned that it has implications for other eye disease. What about just other general inflammatory diseases?
We believe RED1 is important in the context of a number of diseases that are associated with oxidative stress and inflammation, and that's a range of pathologies. We have another talented postdoc in the lab, Dr. Sandeep Subramanian, who's actually looking at this in the context of age-related macular degeneration. And we see a very similar protective effect when we genetically delete RED1 in the retina of mice where we're modeling age-related macular degeneration. So we're hopeful that this has implications even beyond diabetes.
To just go off on that, in the context of inflammatory disease, there has been a number of studies that has implicated RED1 in other inflammatory disease cell types, including like sepsis and IBD and diseases that depend on this recruitment and infiltration of immune cell types. And diabetes is another complication where the immune system is dysfunctional, leading to this overtly immune response in the retina as well as in the kidneys. Some of the research that we have been doing now has been focused on targeting or at least identifying whether RED1 is involved in that pathway and regulating those pathways.
Interesting.
So, Michael, it sounds like RED1 has multiple roles across the body, right? And targeting RED1 may be a viable option for an anti-diabetic therapeutic. Where do you put this in the broader landscape of existing treatments or treatments that are on the cusp of being available?
First and foremost, when we treat diabetes, we're trying to address impairment in glucose homeostasis, and we're doing that by trying to restore insulin action, right? That's not necessarily what we think RED1 is doing. We don't necessarily, in our mice, we don't see any changes in glucose homeostasis. What we see is a reduction in complications that occur in the context of that disrupted glucose metabolism. Like hyperglycemia is still present in our animal models, and the complications are not developing. So conceptually, I think you still, try to address blood glucose changes through, insulin administration and, GLP-1 agonist or SLG2 inhibitors. But what this would potentially do is allow you, to maybe reduce complications when glucose levels aren't being managed as well as they maybe could, right? So if you still have some elevated glucose levels, maybe this could be an intervention to then prevent some of those complications from developing in that context.
It's interesting, right? I mean, it's still a progressive disease. And so being able to treat the complication side of thing is an interesting or hopeful option, right?
Yeah, agreed. It's a very interesting subject. And before we wrap up, Dr. Dennis, is there anyone that you wanted to recognize as contributing to a large portion of this work?
Yeah, so I think it's worth mentioning that a number of these studies that were conducted in my lab were part of a dissertation project by a talented grad student, William Miller, who is actually the one that kind of discovered this redox switch in the RED1 protein and showed that it was acting to reduce the rate of degradation.
Thank you, Dr. Sunilkumar and Dr. Dennis, for joining us today. And thanks to our listeners for tuning in to this episode of Sweet Talk. Be sure to check out their article titled REDD1 Is a Promising Therapeutic Target to Combat the Development of Diabetic Complications, a report on research supported by Pathway to Stop Diabetes, available in the October 2024 edition of Diabetes. And as always, please like, follow, and subscribe for more Diabetes Bio content.
All right, everyone. Don't forget about ADA's special offer for new and renewing members. That's a 15% discount on a 1 or 2 year membership. Go to professional.diabetes.org/membership, create your account, fill out the application, and use the discount code FALL, all capital letters, 24 at checkout. Act fast because this offer expires November 30th, 2024. Don't miss out on this fantastic opportunity to expand your commitment in the fight to end diabetes by joining the American Diabetes Association.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope you are enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournals.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you next month.